Good evening, everyone. My name is Subhashni. Thanks for the opportunity. My research topic is on comparative analysis of clustering methodologies in DNA storage. Well, a person in this year uses 1.7 MB of data per second. We all generate nearly 3.75 into 10 power 18, that is 3.75 hexabyte of information due to the usage of wide variety of billion of devices per day. And it is estimated in 2025, the amount of information required to be stored will be around 175 zettabyte. How are we going to deal with such a huge demand on data storage? Well, nature provides a solution with DNA through its high information density and preservation longevity. But it also has different errors such as insertion, deletion, and substitution affecting the nucleotides, adenine, guanine, cytosine, and thymine. So in order to deal with those errors, the error correction coding is performed, or the error correction coding along with clustering methodology is performed. The main objective of our work is to find out what are the different clustering methods available in both biological and DNA storage domain and modify the algorithm to make it more suitable to the DNA storage domain. And also to evaluate the different performance measure to find out the suitable clustering mechanism, such as recovery rate, sequencing, decoding complexity, and computational time. First, similar to all the different storage devices, the digital files are encoded. Before storing it as a DNA molecule, it is mapped to adenine, guanine, cytosine, and thymine. And then the process of storing happens by synthesizing the data into the DNA molecule and then storing it and then reading the data back through the sequencing and then decoding the data. During this process of storing, the biomolecular errors happen. So these errors can be solved by also another advantage that the DNA storage technology provides us with. It is the amplification of the storage molecules. So when we pass 10 data, it will get amplified to 100 molecules. And out of that 100, we can take only the 50 data. Means I'm taking 5 times of 10 data, which represents as 5 in 5x coverage. And if I'm passing the 50 data to decoder, it increases the decoding complexity. Hence, we are using the unsupervised classification method to get our 10 data back, thereby reducing the decoding complexity. Many research groups work on different types of encoding methodology. Out of that, 2 research groups use the same coding method, and in addition, the Geong research group uses the cooperative sequence clustering method, and they were able to reduce the recovery rate from 7x to 4.77x. This tells that the clustering methodology helps us to reduce the coverage and increase the recovery rate. There are 2 problems in DNA storage technology. The first one is loss and the second one is error. So let's say we are passing 3 data, and after processing it through the DNA storage channel and clustering it, we were able to not achieve the third output, means the third output got lost. But we can get that third output if we set appropriate logical redundancy. But then in order to deal with indel and substitution error, again, the clustering has to be done with high coverage. But then if we are using high coverage, it's not good for the sequencing complexity since the sequencer has to be read a lot. So to eliminate that, we have to design a suitable clustering tool that uses the less coverage value and produce the output with high accuracy. So our major focus is to work on the different clustering methodology. And these clustering methodology works in combination with the algorithm in bioinformatics. They are exact matching and approximate matching algorithm that does the alignment using the different formulas. And some uses the quality value also that represents the probability whether a particular nucleotide is same as the original one or not. So these are the different clustering tools which we are going to modify and evaluate and tune the parameter and find out the best clustering tool. For the slide it uses exact matching. Star code, it uses approximate matching. Mesh cluster uses the combination of two algorithms. So the first is GLM or alignment based approach, and the second is mean shift algorithm. MNS and CSE uses the Hamming distance based approach. So we took the three data set, and these are the different encoded DNA sequences. And out of that, a particular read sequences are extracted, and we are using different coverage value and splitting them into different values, and then finding out whether we are able to achieve the highest recovery rate with the minimum coverage with certain clustering tool. Also, the distance is coming into picture, because when we increase the distance and reduce the number of clusters, thereby increasing the number of members in the cluster, produces the best output with less decoding complexity. And the different algorithms are tuned, like star code, slide sort, MNS, and mesh cluster. For the star code, the message passing is selected because of its highest recovery rate. In case of slide sort, a new post-processing algorithm is designed to determine the clusters and estimate the representative sequence. And for MNS, the parametric tuning is done. In case of the mesh cluster, we work on two modifications. The first is generalized linear model and evaluation of the best fit of parameter. We do the modification on the GLM in order to achieve a high accuracy and to avoid this fluctuating values and increase the speed, providing a good recovery rate. And the parametric tuning is done in addition to it. So from the experiments conducted, the slide sort star code majority nucleotide selection cooperative sequence clustering algorithm produces the best result for all the data set, keeping the fixed coverage value. And star code produces a better output compared to any other algorithm. In terms of speed, the majority nucleotide selection algorithm produces a very poor output compared to the rest because of its environmental settings. In this, the first row represents the information related to sequencing complexity, and second row about the decoding complexity. So let's say I'm keeping the recovery rate fixed to 80%. For data set 1, we require 4.3. For data set 2, it requires 3. And for the data set 3, it requires 2. So which means that star code, MNS, and CSE provides the best result in all the data set, except in case of data set 2, CSE doesn't provide, MNS doesn't provide a better result. In terms of decoding complexity, it has to provide the representative sequence in such a way that it is equivalent to the number of input encoded DNA sequencer. The slide sort provides the best result, but then it is not good since it produces a poor recovery rate. Then comes the MNS and CSE, achieving the comparable decoding complexity compared to the slide sort. Whereas the star code produces more decoding complexity. In terms of clustering distribution, again the star code deviates, providing a high number of cluster with a cluster element which is equivalent to 1. So that again tells us that the star code doesn't provide the good output in terms of decoding complexity. Overall, this is the summary of the experiments done. So the recovery rate is being computed for all the data set, and sequencing complexity is computed with the fixed recovery rate, and decoding complexity with the Hamming distance of 8. From these records, we understood that the star code provides the best lower sequencing coverage with the highest recovery rate. And also the sequencing coverage is quite very low for all the 3 different data sets, which can be seen by looking at the standard deviation, which is of the value 1. But still the star code produces high decoding complexity. CSE requires 0.9x coverage more compared to star code, but it has its dependency with the quality value as well as the Hamming distance. MNS provides the best result with 1.2 coverage more, but then it is quite very time consuming one. So these kind of problems or drawbacks can be rectified in our future work. But the overall, the experiment which we have done help us to give insights of parametric tuning of the existing tool and designing new tools for DNA data storage. We can draw a conclusion that Starcode, CSE, and MNS provides a good performance overall, though it each has its own limitation. The tools and algorithms are evaluated for minimum coverage and maximal recovery rate, which corresponds to low sequencing complexity and high recovery rate respectively. Thank you.